The next part in sexual reproduction in flowering plant is essential whorls. So we have two essential whorls and two non-essential whorls. Non-essential whorls we studied in the previous lecture session, and in today's class we are going to study about androecium and gynoecium, and androecium we will be studying in detail in this lecture session. So what is androecium? It is a essential whorl of stamen. So how do you remember androecium is a male reproductive unit, stamen, androgen, so it's all related to male. So that is male reproductive unit. So male reproductive unit. So why it is male reproductive unit? Because it contains the cells called microspore mother cells or pollen mother cells which contain male gametes. So now if we consider what is the function of this male reproductive unit, it produces microspores, also called pollen grain, and in other terms it is also called male gametophyte. So male gametophyte contains male gametes. How and process we will study in a very short while. So this is about the androecium. Essential whorl of stamen, it is a male reproductive unit. Function of this stamen is to produce microspore or pollen grain, which is otherwise called male gametophyte, which produces male gametes, or it contains male gamete. Next, coming with the gynoecium. Gynecology. So who will go to gynecologist? Women. So it is a part of female. Okay, that's how you have to remember. This is a remember technique I am telling you. So essential whorl of the pistil. Okay, so what is the function of this? What it is? It is a female reproductive unit. What is gynoecium? It is a female reproductive unit. Okay, and what is the function? Very first is it produces female gametophyte, which otherwise called embryo, which is otherwise called ovule, which contains an embryo sac, This embryo sac contains an egg cell plus synergids plus antipodals and central cell. So composition of embryo sac is egg cell, synergids, antipodals and central cell. So we will learn about it in detail in the next session. So what are the other functions? Is it only the function to produce female gametophyte or the ovule? No. It also provides us helps in the formation of seeds. So what are seeds, children? Ripened ovule or fertilized ovule. After fertilization, ovule will transform into seeds. Then the next one is formation of fruit. So what is fruit? It is an ripened ovary, right? Ripened ovary. So this is about the female reproductive unit. What is the function? Produces female gametophyte or ovule, formation of seed and formation of a fruit. So that is with the female gametophyte. The next concept of this chapter is pre-fertilization events. In this topic, we are going to study the detailed structure of stamen, microsporangium and pollen grains. So in the very first, So when we look at a flower, so you just pluck one hibiscus flower or any lily flower or any random flower, you get it in your garden. So observe for the male reproductive structure, female reproductive structure, petals and sepal. When you look at an androecium, so what you will see is a long slender filament and a bilobed structure called anther. A bilobed structure called anther and a long slender stalk-like structure called filament. Right? So in this anther, we see it is a bilobed structure and dithecous. So we will learn what are these dithecous and how it is bilobed in detail. Coming with the filament, it is attached, its basal end is either attached to petal or any other part, say for example thalamus, other part of the flower. So petal or any other part of the flower. Say for example thalamus. As I told, we can write it in a bracket, thalamus. Right? So next, the distal end. So if this is the basal end or proximal end, we can say. Why proximal end? Because it is present near to the parts of the floral parts. Right? So now if we talk about the distal end, what is present? An anther. So it is where our microsporangium is present and pollen grains are produced. So if you look at the NCERT diagram, so the stalk or the filament is long, stalk-like and slender. So it is like a thread, right? It is a thread and made up of an tissue. Now the anther, you can see from the diagram that it is bilobed structure. So anther is present, and the red line what you are seeing here in your NCERT textbook, when you see, it is the longitudinal groove. It is a longitudinal groove. Now let's go with the next concept of studying the anther in detail. So what is anther? As I told, bilobed structure separated by a tissue called connective tissue. Each lobe consists of two sac-like structure which are called microsporangia or sporangium. Okay, so each lobe consists of two sacs. So totally two lobes we have. Okay, lobe 1 and lobe 2. So 2 in 2 sac, right? We have to multiply. Then how many sac will be present? 4 sac. Okay, 4 microsporangium. So hence it is also called tetrasporangiate. Okay, tetrasporangiate meaning 4 microsporangium. So since it is having two lobes, it is called dithecous. The anther is called dithecous. Next topic is structure of anther. So anther is an bilobed structure present at the distal end of the filament. So it is bilobed. Each lobe is separated by a tissue called connective tissue. So each lobe contains two sac-like structures called microsporangium. So it is this microsporangium in which pollen grains are produced. It is this microsporangium in which pollen grains are produced. Since lobes are separated by a septum. Okay, by a septa. Septa means it's a division or compartment. Okay. Now, since we have two lobes, we have two lobes in my anther, and each lobe contain 2 plus 2, right? Each lobe contain one ear in, let us say this is one lobe, this is the second lobe. Here 1 microsporangium, here 1 microsporangium, here 1 micro. Totally how many microsporangium we have? 4 microsporangium. 4 microsporangium. Hence we call anther is dithecous because it is having 2 lobes and also has 4 microsporangium. Anther is bilobed, dithecous and tetrasporangiate. And if you look at the outline of this anther, how many sides they have? 4. So it is also called tetragonal in shape. It is tetragonal in shape. Is it clear, my dear children? Each lobe consists 2 sac-like structure, microsporangium. It is in this microsporangium where pollen grains are produced. Lobes are separated by a septa and each lobe contains 2 microsporangium. So since we have 2 lobes, there are 4 microsporangium. Bilobed because 2 lobes, dithecous because again 2 lobes, and they are separated by a septa, and 4 microsporangium, hence tetrasporangiate. In the outline it is shown as a tetragonal in shape. And if you look at the diagram in the NCERT, okay, so here this groove what we are seeing, okay, will run throughout the length of the anther, So this is the also possibility for line of dehiscence. So why it will help this line of dehiscence? This helps in when the pollen grains are formed and it is in this line, in this groove, the anther will burst out and release the pollen grains, that is when matured. So it runs throughout the anther lobe and this is where the pollen grains are produced, right? So now another aspect of this is this line of dehiscence. Then there should be some walls, right? There should be some walls that we are going to study in the next slide. So what is dehiscence, children? If anybody asks you what is dehiscence, how do you tell? How do you explain someone? So it is the breakage due to dehydration in the walls of the anther. Even fruits dehydrate. For example, you might have seen ladies' finger fruit. After completely grown, after completely mature, they break so that the seeds will go and fall somewhere. In the same way, even anther is also having the walls and they will dehydrate when dehydration. Okay, so that is one thing about this dehiscence. Next, coming with the structure of microsporangium. So microsporangium is present inside a tetragonal structure, but when we look at the diagram in our NCERT or under the microscope, the microsporangium looks almost circular in shape. These are microsporangium and circular in shape. Is that clear? Now, why studying of the structure of microsporangium is important for us as student, because for 5 marks this question is asked for boards. Draw a neat label diagram of TS of anther. How you will write? There are 3 diagrams, 1, 2 and 3. The first 2 diagrams we need to write, and I will show you now how easily we can write the diagram and also understand the detailed structure of microsporangium. So in a TS of anther, when we observe under microscope, the microsporangium almost appears circular in shape or outline. So how do we draw the diagram? So first, what exactly microsporangium contains? It consists of walls, it consists of sporogenous tissue. Sporogenous tissue. So how it is arranged, we will study. Okay. So I'll take up a new slide for that. So first, how do you draw when it is asked in exam? How do you practice it? Okay. So first, write a bigger butterfly like this. So we write butterfly in the same pattern, right? So write like this. Okay. Then fine. Now write 3 walls of same kind. Okay. So this comes our epidermis. Walls we said, right? Epidermis. Then comes endothecium. Endothecium. Then comes middle layer, then comes middle layer, then comes, now you draw 4 circles in this corners. That is our microsporangium. That is our microsporangium. And around the microsporangium, draw some cells. Okay, like this. Okay, draw some cells. So in textbook it is not there. So let us, okay, we will do one thing. So we will stop it for this one. We will go per as per NCERT text. So this is the young anther, and here we have to show the connective tissue. So what is this one? This is the connective. Okay, connective tissue. So this is the TS of anther, young anther. Okay. So now we have to just take this part, okay, and enlarge it and write. So how do we write?